Hello, students. And this is the topic made for the year 2020. Oh, yeah, you've been waiting for it since September. Now it's time to unpack it in all of its splendor and glory. What topic am I talking about? Well, that topic is viruses and bacteria, the talk of the country and the world for the year. And so we are going to be focusing on viruses and bacteria this week and performing a little lab activity, a modified activity on a Thursday. So why don't we get started right away? So let's begin with the first part of our topic, which is Kingdom Monera. Kingdom Monera is the fancy name for this kingdom, and you can see that picture of petri dishes. We'll be talking about petri dishes later on today during this lesson. But Kingdom Monera is made up of organisms called monerans that we know them better as bacteria. Bacteria are also called monerans. Monerans are also called bacteria. And Kingdom Monera is made up of organisms that we generally refer to as bacteria. Now, the lab activity this week has to do with collecting samples of bacteria and growing them and then examining them under magnification and, well, examining even under non-magnification, the colonies of millions and millions of bacteria. Before we even consider, however, performing any type of an experiment with bacteria, we have to talk about a few things first. So, first things first. Whenever thinking about collecting bacteria, we have to remember that they are all around us and in us all the time. We cannot escape bacteria. There are good bacteria and there are bad bacteria, but there are bacteria growing on us, in us, around us, between us. Bacteria thrive on planet Earth. And so we have to remember that there are bacteria all over the place. No, we can't necessarily see them without magnification or without growing a colony of them, but you have to be aware that there are bacteria all over the place. That's one of the reasons why it's so important to wash your hands all the time, especially before mealtime, because if you don't, there could be harmful bacteria on you that you would be consuming, and that could potentially be a bad thing. So, item number one, they are all around us all the time. As I mentioned already, some of them can be dangerous. There are deadly bacteria surrounding you right now. Now, don't be scared. The human body is designed in such a way with a fabulous immune system that more often than not, we are protected against dangerous bacteria. And that's a good thing. Our bodies have various lines of defenses, including our skin, our first line of defense. But we've got, assuming we're healthy, we've got good immune systems to ward off the introduction of dangerous bacteria into our bodies. But we have to keep in mind that there are bacteria in this world that are deadly and can kill us. And so whenever we consider collecting bacteria on purpose and growing them, we definitely have to keep in mind that there is a potential danger, so we need to be careful, and we will take precautions to avoid any bad things from happening to us. Number 3, because bacteria are in fact microscopic, they are unicellular organisms, but they live in colonies. In order to observe them with the unaided eye, in order to observe them with the unaided eye, we need a colony of them. So we're going to be growing colonies of bacteria in the laboratory. That means we're going to be providing a suitable habitat for millions and millions of them to flourish. More on that in a little while. Number 4, we can incubate them in agar. Now, there's a couple of words there. What the heck does that mean? Agar is a nutrient-rich food that bacteria grow on. So we're going to be providing a food source for our bacteria, and that food source we're going to put into things called petri dishes. And then we're going to keep them incubated or keep them warm. We're going to keep them at a particular temperature for a particular amount of time. And given the right conditions, they will be able to flourish, meaning reproduce and grow more and more, which means we must make sure that we seal shut the petri dish. Once we collect our samples of bacteria, and we put them into petri dishes, and we put them into an incubator, we don't want them to escape. That would be bad because, as we know from number 2 here, some of them are dangerous. So we don't want to come into contact with millions, if not billions, of bacterial cells if they are dangerous to us. So once we collect our sample, these petri dishes will be sealed shut so that they will never be opened again, at least not by us. Now, here's an interesting thing to keep in mind when storing away our petri dishes in the incubators. The bacteria that we will be collecting, well, just like us, we could drown in our own wastes. That's a disgusting thought to consider. But bacteria will drown in their own waste. And for the first part of this activity, we don't want to kill them right away. We want them to flourish. So in order to prevent them from drowning in their own waste material, we store the petri dishes upside down. Let me try to explain that here. Here we've got a petri dish, and in the bottom of the petri dish, there will be what is called the nutrient agar. We'll collect the sample, and we'll put samples of bacteria into the bottom of the petri dish. Then we put the lid on top of it and seal it closed with tape. Now, if we just put the petri dish like this into an incubator, it's warm in the incubator. And any waste material that is produced by the bacteria will evaporate. It'll create a little weather cycle inside the petri dish. And a little vaporized waste material will rise to the top of the lid, which will be a little bit cooler, will condense like a rain cloud, and then precipitate down. It will rain liquid waste on top of our colony of bacteria and kill them. We don't want that to happen, so we will store our petri dishes in the incubator upside down. So they'll be growing upside down so that all of their waste will be carried down by gravity and they won't drown in their own waste. Isn't that a lovely thought? So here's a picture of the agar in the bottom of a petri dish, and a petri dish has a lid. The lid will go onto the bottom of the petri dish. The whole thing will be flipped over and stored for a few days in the incubator. And once that is done, then later in the week on lab day, we'll actually remove them from the incubator and we will be able to compare the various samples in the petri dishes. Yum, yum. Okay. More on that later when we talk about the lab activity. All right. So let's move right on then into Kingdom Monera. So let's learn a little bit about this kingdom before we start going around and collecting them. Okay. They are single-celled organisms that lack a nucleus. What? Wait a minute. You told us, Mr. Skirps, last unit that all cells have a nucleus. What's up with that? Well, I may never tell you the whole story, because if I told you the whole story on every little piece of information, I would be your teacher for the rest of your life, because there's so much more to learn. But right now, I'm telling you more of the story. Bacteria or monerans are simpler organisms, more specifically called prokaryotes, that lack a nucleus. Now, that doesn't mean they lack genetic information that other cells have in their nucleus, but they just don't have a structure, the organelle structure within each of their cells that could be called a nucleus. So, yes, monerans have no nucleus. They have no brain. They are like the scarecrow in The Wizard of Oz, you might say. But be that as it may, they are living organisms, and they deserve an incredible amount of respect, because bacteria have killed more human beings than anything else on planet Earth during human history, believe it or not. So let's learn a little bit more about these monerans, also known as bacteria. So let's review a diagram now of the typical structure of a bacteria. But keep in mind that this diagram is a typical structure, which means there's a lot of variability. There are many, many, many differences between bacteria. There are loads of different species of bacteria, but most of them, by and large, have some similar structures. So in your books, and let's see, in your books, you have a diagram that looks like this, and I would like you to label your diagram. Let's start with the inside and work our way out. These little squiggly lines here, this is called the hereditary material. These are the chromosomes that are made up of the DNA that determine what kind of bacteria this is. And they're kind of floating around in the cytoplasm, the jelly-like material that surrounds them. So even though the hereditary material isn't contained within a nucleus, and by the way, some of them actually are contained in something called an endospore, like a baby nucleus, but we'll talk more about that later. They all still have hereditary material, or they wouldn't be able to reproduce. So we've got the hereditary material surrounded by the cytoplasm, and then we've got different layers here. We've got the cell membrane right around the cytoplasm, around which is the cell wall. And the entire structure is called the capsule. And at the end of the capsule are these squiggly lines, which are called the flagella, which are whip-like structures that can help some bacteria move from one place to another. Not all bacteria have flagella, but those that are mobile do or can possess flagella, which, by the way, is the plural form of flagellum, which is singular. Now, when I look at this diagram, I see something with my imagination. And for the quiz later this week, you might have to reproduce this diagram. You might have to draw a diagram of the typical structure of a bacterium. And the way I see this is it looks like a slipper to me. See if you can picture it, because I want you to keep in mind that these structures are three-dimensional. They're just not flat diagrams, they're three-dimensional. So imagine this is where you put your toes, and this is where you put your foot. And these squiggly lines are like the little massage of a little foot insert, perhaps, or shoe insert. And the little squiggly lines are like the stinky odor from your feet between your toesies, maybe from that like that diagram we saw last week, if you remember, from the fungus, the foot fungus. But that's what I see. I see a slipper here that you could slide onto your foot. You can also view it. I can also see a jelly bean that has been dissected. The jelly bean has been cut in a quarter. A quarter of it has been removed, and you're looking inside the jelly bean. In either case, I want you to try to remember that bacteria, just like anything else we would view under the microscope, are three-dimensional objects. And so they have dimension, they have depth, they pop out. They're not just flat. All right, now I mentioned a word here a moment ago called an endospore. Some bacteria, more developed bacteria, do have a structure inside where the cytoplasm is, called an endospore. And the endospore is where that hereditary material can be found. Now, it's not a true nucleus per se, as in other cells, but it is kind of a primitive nucleus. Now, the word endospore, if we break that word apart and dissect it, the prefix endo means literally inside. Spore literally means seed. So the word endospore literally means inside seed. And as I mentioned, some bacteria, not all, but some bacteria have this structure, an endospore, with hereditary material inside it. The endospore can provide another measure of protection for the viability of the hereditary material within its structure. But as I said, not all bacteria have an endospore. So some bacteria have it, the endospore, with hereditary material. Now, I mentioned earlier that there are many different varieties of bacteria, and there are. We certainly have not yet discovered all of the bacteria on planet Earth. I would imagine scientists are exploring or are discovering new varieties probably on a daily basis, especially now with a greater emphasis on the microbiology world and so many people studying these tiny little organisms. We'll probably have additional groups in the future than we have now. But broadly speaking, bacteria are divided into three main groups based on their shape, their morphology, their outside structure, their phenotype, you might say. So the first group of bacterial shapes are spherical. But of course, scientists can't use words like spherical, so we use words like cockeye. Cockeye is the correct term for spherical bacteria. So if we examine bacteria under the microscope and we get a closer look at the bacteria that we may be collecting this week, we may see that some bacteria have a round structure. And that round structure, there we go, look at that. A green ball. Isn't that cool? This round structure, if we see bacteria like that, we will call them cockeye. Now, the various varieties and species of bacteria If they are spherical in shape, they will all have cockeye as part of their name. Now, one way I use to remember that cockeye are spherical is look at the word cockeye. C-O-C-C-I. The letter O is spherical, like a ball. It's spherical, like a ball. The letters C, C, and C, they're all somewhat spherical also, like a ball. The dot on the I is spherical. So you've got 1, 2, 3, 4, 5 hints in the word cockeye to relate it to the spherical shape. All right, that brings us to the next shape, which are bacilli. Bacilli. Bacilli aren't so silly if they get you sick, but bacilli are rod-shaped bacteria. Here I have a Sharpie, a green Sharpie, by the way. Ooh, look at that. It looks black there, but it's a green Sharpie. Let's see if I can do anything fun with a green Sharpie here on the green screen. Oh, right into my mouth. All right. Green Sharpie. If you look at the shape of a Sharpie, it's kind of like a rod. And if we examine bacteria under the microscope and we see bacteria that are more or less rod-shaped, they could be bent, of course, but they will undoubtedly belong to the group of bacteria called bacilli. Now, there are a couple of hints that I get from the name bacilli. I think of the letter L. L. 2 letters. L. L. L have a similar shape as a rod if it's going up and down. At least that's the way I see it. So when I see bacilli, I see 2 rods on bacilli. That's how I remember that bacilli are rod-shaped. And that brings us to the third category called spirilli. Spirilli. Spirilli, I think, is just kind of easy to remember because spirilli bacteria are spiral-shaped, and spiral and spirilli are very close to one another. And if I think of spirilli pasta, I think of little twisty turns, little spiral pasta. I also think of slinkies. And here's a green slinky here, which may make it fun to see on the green screen. But a slinky is a spiral shape. And this spiral shape we might see in the shape of various bacteria. And if they're spiral shaped, well, we will call them spirilli. So spirilli are the spiral-shaped bacteria. So we've got lots of there are many, many different kinds of bacteria, but 3 main shapes of bacteria: cocci, bacilli, and spirilli. And so I think with that information in mind, I think we will call it quits here for today's video. I'm going to give you some time today to watch some interesting videos on bacteria and kingdom Moneran. So we'll just call it quits here for today. You watch the videos, and I will see you tomorrow with the life functions of bacteria. So for now, bye-bye.